Romans in Britain were more diverse than we thought. DNA rewrites the empire. When we imagine Roman Britain, we usually picture pale European soldiers marching through cold northern landscapes, bringing Roman roads, Roman law, and Roman order to the edge of what they believed was the known world. We imagine forts standing against endless fog, legions defending the empire from distant barbarian tribes, and Latin echoing across damp stone walls. But what if that image is incomplete? What if many of those soldiers were not born in Europe at all? What if Roman Britain was home to people whose ancestors came from Africa, from the Middle East, and even from regions far beyond the Mediterranean world? This is not modern speculation. This is not political reinterpretation. This is DNA, extracted from ancient graves, tested in controlled university laboratories, and published in peer-reviewed scientific journals. And what it reveals is forcing historians to rethink what the Roman Empire truly was, not just as a political power, but as one of the largest population mixing systems the ancient world had ever seen. What history long believed. For centuries, almost everything we knew about Roman Britain came from Roman writers, military documents, and archaeology. These sources told us where legions were stationed, where forts were built, and which emperors controlled the province. But they rarely told us where individual people actually came from. Gravestones listed ranks and units, not childhood villages or ancestral homelands. Archaeology uncovered helmets, shields, swords, and coins. Latin inscriptions appeared everywhere. Roman-style villas and bathhouses reshaped the landscape. From this, historians naturally assumed that culture meant Roman people. If the buildings looked Roman, if the army followed Roman rules, then surely the population must have been largely European and Mediterranean. But culture spreads faster than people. An empire can export language, laws, clothing, and architecture without moving entire populations at the same scale. Without genetic evidence, there was simply no way to see beneath the armor, beneath the pottery, beneath the inscriptions carved in stone. So for generations, Roman Britain was imagined as mostly European, with only rare exceptions. That assumption would not survive modern science. How universities study ancient genomes. Everything changed with the rise of ancient DNA research. Today, in sterile, clean rooms at universities, scientists wear full protective suits to prevent modern DNA from contaminating ancient samples. They extract genetic material from the densest bones in the human body, especially teeth and the petrous bone of the inner ear. These bones act like natural safes, protecting fragile DNA fragments for thousands of years underground. The DNA is usually damaged, broken into tiny pieces, and chemically altered by time. Using specialized enzymes and chemical processes, scientists repair and copy these fragments millions of times through amplification techniques. High-throughput sequencing machines then read each genetic letter, reconstructing large portions of the ancient genome. That genome is compared to massive databases of both ancient and modern populations from Europe, Africa, the Middle East, and Asia. From this, researchers can identify ancestry clusters, migration histories, and sometimes even family relationships between individuals buried in the same cemetery. But the process does not stop there. Before any results are accepted, they undergo rigorous peer review by other scientists. Data is rechecked, contamination is ruled out, and interpretations are carefully tested.
Only after this long process are the findings published. Often years pass between the excavation of bones and the final scientific conclusions. This is slow science, but it is also extremely powerful. The results that shocked historians. When graves from Roman Britain were genetically analyzed, the results were not what historians expected. Some individuals buried with Roman military honors showed genetic ancestry tracing back to North Africa. Others showed connections to the Eastern Mediterranean and the Near East. In several cemeteries, entire family groups shared mixed ancestry patterns, suggesting long-term settlement rather than temporary deployment. These were not isolated travelers, they were communities. One of the most famous discoveries revealed ancestry linked to sub-Saharan Africa in a woman buried according to Roman customs in what is now England. She lived, worked, and died in Roman Britain, not as an outsider, but as part of local society. These people were not symbolic representations of diversity. They were ordinary residents of towns and villages, raising children, forming families, and aging in place. What historians once saw as a distant frontier outpost now looked more like a crossroads of the empire. The Roman Empire was not just conquering territory, it was redistributing people across continents. Why the empire was so genetically mixed? This diversity did not happen by accident. Roman military policy actively encouraged movement. Recruits were taken from across the empire. A man born in Syria could be stationed in Britain. A cavalry unit of North Africa could guard the northern frontier at Hadrian's Wall. Service lasted decades, and after retirement, many veterans were granted land locally, giving them strong reasons to settle permanently. Merchants followed the army, supplying food, clothing, weapons, and luxury goods. Where soldiers went, markets and towns soon followed. Families migrated together, especially along major trade routes connecting ports and forts. Over generations, marriage connected local populations with newcomers, blending ancestry across social classes. Britain became genetically connected to Africa, the Middle East, and Southern Europe, not through mass invasions, but through daily life, commerce, and service. This was not forced displacement alone. It was opportunity, security, and citizenship that encouraged people to move. The empire functioned as a massive migration engine, constantly reshaping the genetic map of its territories. How this changes modern history education. These discoveries have transformed how ancient history is taught. Archaeology is no longer enough. Today, research teams combine multiple sciences: genetics to track ancestry, isotope chemistry to identify childhood environments, burial analysis to understand social status, and historical texts to provide political context. This interdisciplinary approach has rewritten major historical assumptions. Migration is no longer seen as rare, it is recognized as constant. Ethnicity is no longer treated as fixed, it is understood as fluid and overlapping. Students now learn that ancient societies were dynamic systems shaped by movement, not static populations isolated by borders. Empires did not just spread ideas, they physically relocated millions of people across vast distances. Roman Britain is now studied not as a peripheral colony, but as part of a continental population network. What DNA really says about human identity. One of the most important lessons from genetics is what it does not tell us. DNA does not define culture. A person could speak Latin, worship Roman gods, wear Roman clothing, and obey Roman law without having ancestors from Italy. Identity is learned, loyalty is chosen, belief is cultural. DNA only records where ancestors once lived, not what people believed, not which empire they defended, and not how they understood themselves. Roman Britain was not divided into Roman and foreign. It was a blended society where identity was social, not genetic. Modern ideas of race do not fit the ancient world, and DNA is helping historians finally see that. Final message: why this matters. The story of Roman Britain is not only about ancient diversity, it's about how easily we mistake culture for blood. Long before airplanes, passports, and nation-states, people crossed continents through empires, trade networks, military service, and survival. Migration is not new. It is one of the oldest human behaviors. DNA does not